Hey guys, this is Mr. Kelbley. Today we are starting a series of video podcasts. For the most part, you'll be watching these at home, but for this first one, I wanted to view it in class so that we would all get an understanding of what this would look like. So today's topic that we're going to be talking about is transcription and mRNA. Some of this will be a little bit of review, some of it will be new, but it's all going to be important for you to know. So just a brief note on how these videos will work. It's just like taking notes in class. So I will have boxes or blanks for you to fill in on a worksheet. And what you're going to need to do is, just like in class, anything that you see that is purple, underlined, italicized, and bolded, you'll need to write down. So anything that looks like this right over here. The cool thing about doing notes this way is that you'll be able to pause and rewind, anything like that if you need to, so that you can learn at your own pace. That way we won't be waiting for some people to take notes. Everyone can go at their own pace. Usually there will be questions to answer at the end of the video as well, although that will not be the case today. Again, the whole point of this is so that we'll have more time in class to do fun things like projects, like experiments, and so we can get a little bit of note-taking out of the way at home. So again, today we're talking about transcription, and I just wanted to review, to begin with, what transcription actually is. This is the process when information from DNA is converted to RNA. And as you follow along in your guided note sheet, you'll see this is already filled in. What's not filled in is this next one. The question is, where does transcription take place? The answer is the nucleus of the cell. Because remember, DNA is in the nucleus. So that's where transcription has to happen. So moving forward, what is RNA exactly? We've already talked about DNA a lot, but we haven't really talked about RNA so much. A couple key differences that I want to make sure you are aware of. First of all, RNA is just one single strand, whereas DNA, as we all know already by now, is a double helix. The second one involves nitrogenous bases. Remember, DNA has 4 nitrogenous bases, A, T, C, and G. RNA also has 4, but the main difference is, instead of T, RNA has U. So what this means is RNA has A, U, C, and G. A and U will always pair together. C and G will always pair together. It's just how it is. RNA has a different molecule, U, instead of T. So that's a little bit about the basics of transcription. How exactly does it work? Again, you read about this for your notes before, but just to briefly review, in transcription, DNA separates in kind of a bubble. So not the entire double helix, but a little bit of the DNA separates. After this happens, one strand is used as a template for the RNA. So in DNA replication, for example, the DNA would unzip and then both strands would make new DNA. That is not the case for transcription. For transcription, only one strand is the template. And again, as we mentioned before, in RNA, A will pair with U, C will pair with G, because there is U instead of T. That is the only difference there. So here's a picture of transcription that I hope will help you visualize it a little bit. This is that bubble I was talking about, where DNA partially pulls open. What happens then is the enzyme RNA polymerase, which, if it's not written on your notes, don't worry about it. But it'll come in and it will help build this RNA strand. You can see C goes with G again, U goes with A instead of T, U goes with A, then A, T, G, C, all this stuff. You might wonder why you see A going with T here. That's because the T is part of the DNA. This is the RNA that's being built here. So as you can see, the RNA strand is being built out, and eventually the RNA will leave, and it will go to the next step for making proteins. So what comes out of transcription? We talked about RNA, but specifically coming out of transcription, it's called mRNA. The M stands for messenger. The messenger RNA leaves the nucleus after transcription. It leaves the nucleus so that proteins can be built. And we're going to learn more about that step later on. So mRNA, a little more about that. mRNA, it holds the instructions from the DNA, right? Because if you look back at that picture that we had discussed, the DNA is used as a template for this mRNA. And mRNA is divided into groups of 3 called codons. So every 3 base pairs will be a codon. So for example, maybe it will be AUG. That would be 1 codon. And that's something we will be learning a lot more about in the near future. Codons, again, are used to build proteins later. So the mRNA, again, we're going to learn about how this happens in a little bit, but it happens in a step called translation. The information from the mRNA is used to build proteins out in the ribosomes.
What you are about to see is DNA's most extraordinary secret: how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.